today I'll be talking about this CRAWDAD paper characterizing cell type spatial relationships across length scales, spatially resolved genomics data with the DLI here. And basically they present an open source R package called CRAWDAD, like I mentioned. And to summarize it very briefly, it's sort of about quantifying spatial relationships between cell types. So they have sort of a metric for the magnitude of these relationships. It's actually two-directional. So if you have a pair of cell types, there can be sort of a different relationship between using one as the reference versus the other. And then also the spatial scale. So that I think that's one of the big points here, is that they make a point to say that the spatial relationships, so in order to define a spatial relationship, you have to first define the spatial scale at which they occur. I'll get more into that when we cover some of the figures. So they first introduce this package on simulated data just to explain sort of how it works, but then they go on to apply it to a bunch of different real data sets with different spatial technologies. So yeah, first just to give an overview of like how they describe this method. They present their first figure on this simulated data set, basically shown on the right here, where we have 4 different cell types in this it's an artificial spatial pattern, but it's an important one to show how things work. And so we have like certain cell types much closer to others, and depending on the scale that you look at things. But this sort of gets into one of the points with, there's this z-score metric that they compute. So I'll get into sort of how they do that. But the basic question that this slide is sort of answering is that, like, if you have a reference cell type, you look at the neighboring cell type proportions, and you want to see how much those diverge from a sort of a background when you do this shuffling procedure. So to explain a little bit more, in the figure C here, we're looking at cell type B as the reference cell type. So that's this little light green cell type here. So they highlight one cell, but basically we're looking at a neighborhood of each of these cells. So they show one of them at a fixed radius that you can specify, usually fairly small compared to the rest of the tissue. And basically we're taking the union of all of these neighborhoods. So that's illustrated in this yellow circle. It's not a perfect circle because it's the union of many different spaces. So it's not quite guaranteed to be a circle by any means. But so then once you have that neighborhood, that union of neighborhoods, really, you can look at the cell type proportions of the other cell types other than the green one. And then you can sort of compare that to what's done in G, where for a given spatial scale, typically when you run this package, you'll use multiple spatial scales, but we're doing this one at a time. So if we have one spatial scale, you sort of form this grid of squares where the side length of a square is that spatial scale. And then within each of those squares, you're actually randomly shuffling the cell type assignments of each of the cells that belongs in the square. And the idea is that this sort of gives you like a random background about what cell types belong in a neighborhood of a particular spatial scale. And then they do this, I won't cover too deep on the methods, but they use this binomial proportion testing framework. So that returns a z-score ultimately. And the z-score is an indication of how much the proportions found in the neighborhood in C diverge from what you would expect based on this null background, essentially. And so that sort of like gives us a little background to understand some of the next parts of the figure, where we see these graphs in E, z-score versus the spatial scale. So in the previous slide, I had shown just one spatial scale, but they typically you want to run this at many scales, and then you see how this metric sort of changes across the scale. So again, we have this is our reference on the left. So the top and panel E here, we'll try to understand. So if we use this reference cell type of C, which is this light blue, and looking at neighbors of B, these are cell types that are very localized, just looking at it by eye. And so if you imagine those grids from earlier, if we shuffle a very small grids, it's not going to make much of a difference in terms of like the neighborhood versus the null background. It only really becomes evident as you raise the spatial scale. So if we were to think of sort of an extreme case, if you take this whole tissue section, and then you shuffle all of the cell type assignments randomly, so you get this really even colored background. If we're looking at the neighborhood of the reference, cell type C, obviously there's more green in this section than there would be in a random background if you mixed every cell type. So the way that shows up in this plot here is that as you increase the spatial scale, it becomes more and more evident that B and C are co-localized, I guess. And typically these z-scores, one of the main plots that I'll show in a bit to summarize these z-scores takes a single number from these plots, essentially. And that's the number, the first spatial scale at which the z-score becomes greater than, has a magnitude big enough to be considered significant. So there's this cutoff here, which is shown by the dotted line. And essentially it's showing that like even at a very small spatial scale, it becomes evident that these cell types B and C are co-localized. So that's, I guess, the summary takeaway from that plot. We can try this a little bit with a more complicated example where we use the reference cell type A, the red cells, and we're testing neighbor B, so the green. So this is a little bit more difficult to interpret, but if you try to imagine grids of various sizes, if we look at the neighborhoods of this red cell type, there's some green ones nearby and there's some blue nearby. And actually there's really all three cell types are sort of in a neighborhood of these red cells. So even if you shuffle sort of a square, it's kind of hard to picture these grids, but if you were to shuffle the cell type assignments, a grid of a sort of a moderate size, it's not going to make much of a difference in terms of how many green cells are nearby the red ones. It actually goes negative until about 500 microns. But basically, once you do that, what I was describing earlier, where you just shuffle the entire tissue section, just sort of indicating what would show up in this plot on the very right of the highest spatial scales. That's when it becomes evident that A and that there are a lot of the cell type B around A in this relatively small spatial scale, a little bit bigger than what we see on the top plot. But that's why, so the interpretation of this plot is like once the significance only occurs at a much higher scale than what we see in the top plot. So there's just sort of two examples to sort of show what this package is measuring and some of the things that we would typically plot if we were applying this package. Starting with that, so we have these same plots from before on the left. And then, like I said, typically we'll summarize this into this dot plot. And so, like I said, it's like the first spatial scale that reaches the significance threshold. So from the top plot, that's like around maybe 100 microns or so. And if we look at the scale, that shows the size of these dots here. So the largest sizes are around 150 microns. So that plot corresponds to a large circle. It's red because the z-score is positive. In other words, there is some sort of co-localization type behavior. Again, this is sort of a one-way thing. So you can notice that this is using reference C and neighbor B, but there's also reference B and neighbor C, which in this case is a very similar relationship. I will get to some cases where some more complicated cases, but that's sort of where we get this dot here from this top left plot. Similarly, for the reference A and neighbor B, like I mentioned, the scale at which it becomes significant is much larger, maybe around, let's say 800 microns here. And so it's still a positive relationship, right? The z-score is positive, so it is sort of a co-localization type behavior. And so we get a red dot since it's co-localized, and then a very small dot because it's a larger spatial scale. So we have some relationships that are only evident once you look at a larger scale. So this is probably, I think this is maybe one of the more interesting great summaries of what this package can do here. I do want to get into a more complicated example just to really make it clear, because when I was reading this paper, it took a while for me to really click about some of the more complicated examples where maybe if you switch the reference of neighbor, you get different behavior, or even sometimes if the reference and the neighbor are the same, you can even have a negative z-score, which I think I cover soon. But in any case, for this slide, we're talking about a case where switching the reference of neighbor gives you different results. So one of the cases where that can happen is we're checking out in E and F, we have these two different cell types. I won't get into too much of biological context, but just to motivate the sort of what we're observing on a technical level. What's interesting about the relationship between these two cell types is the light blue cell type is sort of scattered throughout the tissue. So there's plenty of regions for this cell type where it occurs at a sort of very separately from the darker blue cell type. But if you were to just use the dark blue cell type as the reference, as this panel G here, you see that the dark blue is, everywhere the dark blue is, there's sort of light blue nearby, right? So that shows up as this positive z-score relationship across many different spatial scales when you're using the dark blue as the reference. On the other hand, like I mentioned, since the light blue occurs at so many places in tissue, many of which occur without the presence of the dark blue cell type, we actually get a very negative z-score relationship across the spatial scales. So the reason that makes sense is because so much of this cell type exists far away from the sort of less than you would expect at random relative to this dark blue cell type. So yeah, so this is just a case where maybe like the spatial relationships can be somewhat complicated. It's not quite fair to interpret. If you have a pair of cell types, you can't always just say they're co-localized if one of the directions of the relationship has a positive z-score. So it can be a little bit more complicated. So I guess that's just like a caveat. If we use this package, we'll have to just keep in mind how to interpret some of these more complex relationships. So this is one of their supplementary figures. And then, so there's a number of real applications that they show in this paper of this method. One of them I'm going to highlight today is a Xenium dataset. I think it's sort of closer to we're maybe a little bit more familiar with Xenium, although we don't do it a huge amount. Basically, this is an example where they have a lot of different cell types. And Xenium, of course, is another technology where we have spatially resolved cells at a very high resolution. So checking out this heat map again, this dot plot of all these cell types, something they do here is they actually sort of cluster, or they order the rows and columns of the heat map to show groups of cell types that are sort of mutually co-localized or mutually dispersed. And so on panel C on the right here, they have 3 different examples of groups of cell types that are all co-localized in this case. So the first example has 4 different cell types. So every single relationship here is red, indicating co-localization. Again, you can interpret the diagonal here as a cell type in itself. And so, like, I guess where I'm highlighting with my cursor, there's this cell type GCIS1, this myo-epi cell type. And if you sort of switch which one's the neighbor in the reference, you still get a red relationship of a similar spatial scale. That's true of each of these cell types here. So that's something useful about the way this clustering works with this dot plot. So, but in terms of what it spatially looks like, you can see that basically it's like a few different cell types that are, of course, obviously co-localized. And you can see sort of, I guess there's a bunch of different ways that this might look spatially, but this is one of the cases here. Similarly, this is a cancer data set. So the next one is an example of like 2 different types of tumor cells. It's a little bit harder to see in this one. You sort of only see the invasive tumor cell type here. I was trying to find some of the purple here. It's a little bit harder to see, but I think that might be just because they're very like interspersed based on the co-localization relationship. Hard to tell here. And there's an example of a huge number of cell types really. So I didn't even show the colors here because there's so many of them, and I don't know if we would really be able to individually interpret them, but you do see sort of these interesting spatial patterns of like small regions where you have the cell types mixing, pretty small spatial scale in most cases. So it's always good to check what these cell types actually look like spatially when you identify that they have this type of relationship based on the package. So this is a cool example just to mix real cell types in a cancer data set. Another figure I wanted to highlight was they do this analysis where they describe how to integrate multiple samples together. It's maybe different than what our application would be. So oftentimes we'll have like a brain region, an individual brain region where we try to get a very similar location in the brain, and we have multiple samples. In this case, they had multiple samples across a given location, but basically like this is a mouse brain, and I think they get sort of a much broader set of regions that they describe as locations here, locations 1, 2, and 3. So it's maybe a little bit different than our normal experimental design, but this is the type of analysis that they sort of showcase. So yeah, and you can see all the different cell types that change as you go across locations, just very obviously by eye, you can see those relationships. But in panel B, they try to summarize sort of the variability of the cell type relationships. And I think eventually, my interpretation from what I remember is that they take the z-score plots that I was showing earlier, and they summarize that as sort of an area under the curve. And they do that as a single number for each cell type pair. They ran PCA on that space, and they are basically just trying to show that like most of the variability that you can see from PCA shows up between locations rather than between replicates within a location. That's something that they show in multiple different ways, but the first one is through PCA. Yeah, and then they go on to show several other types of plots that are essentially summarizing the same information. The variation of the z-score trends is greater across locations than it is across replicates, which makes a lot of sense. So in panel C, they have each cell type pair. And again, they did that area under the curve of these z-score plots to summarize it as a single number. And then they computed the variance across those. For the top one, it was across replicates. And you see these very small variances compared to when they did the variance of those area under the curve numbers across locations. So they highlight a particular pair of cell types, so excitatory neurons neighboring GABAergic neurons. So on the top plot in panel D, we can see this is just one location, and we have multiple replicates. So all three of the replicates show like pretty similar z-score trends. If you contrast that with the bottom plot here, it's the same pair of cell types, but now we're taking one replicate from each of the locations. One of them is even sort of positive, maybe not significant. They don't show the significance lines in this plot, but there is a lot more variability here. And so, I mean, this is like one way to summarize, to interpret maybe if you have this particular type of experimental design with replicates, but maybe not super, it's not maybe completely applicable to when you're trying to find similarities, maybe if you have the very same identical brain region or something, and you have multiple samples that are trying to show similar biological patterns. But this is just their multi-sample example, I guess. So finally, for that pair of cell types, the GABAergic neurons and the excitatory neurons, basically like the variability across locations shows up spatially, of course. So basically in panel E, we have, if we're looking at these middle two plots, sort of, this is the same location and different replicates. So not much difference there, right? So the spatial pattern is not like obviously very different. Whereas across locations, the difference in these z-score trends is also reflected in sort of a different spatial organization of the GABAergic neurons relative to the excitatory neurons. So yeah, I guess this is something that could probably show up in many different ways, but basically it's reflecting a different spatial organization of the tissue. So the manuscript covered a few different examples that I didn't show today. They covered a bunch of different spatial technologies, including slide-seq v2, seqfish, and codex. So we had covered Xenium, and I guess I didn't mention that this was a merfish data set here. And basically this method is applicable when all it takes as input is basically the spatial locations of cells and then the labels for the cell types. So I've been trying this out with Visium HD, and I think like really if you have sufficient resolution where you can get individual cells, this CRAWDAD is going to work pretty well. So there's a lot to explore here. And yeah, that's all.